Welcome back to this class. Hey guys, today we are going to be looking at the prediction, or what do you expect to find in paper 1. You're gonna write. We have been having a very big journey from the beginning of the year. Now it's time for us to end the journey, and we must end the journey with a distinction. That is supposed to be like that. We must end our journey with a distinction. If the distinction is not yet there, use this video so that you can still improve your marks and score high marks, which are going to take you to the university. It is M.SAIDI as usual. Don't forget to share the video so that even others can benefit from our discussion today. Let me know if you have any challenge. Yes, let me know. I'll try to fix it out. I'm gonna make it a little bit longer, maybe an hour. Maybe an hour, so that at least if I don't go that into much detail of some things you have asked me, then maybe an hour will be enough. All right, guys, don't forget that in this paper you're gonna write, yes, you have the first topic as reproduction in vertebrates. If you look at our distinction material, this is the topic I'm talking about, reproduction in vertebrates. So what are you supposed to look at here? Remember, this is a topic which has very few marks, and these marks are supposed to be all of them. Please, when you come here, you need to know what is called fertilization. In this case, it can be external or internal, but don't forget, I'm going to be highlighting the small mistakes students do in each and every topic, so that you don't make these mistakes again. Don't watch the video only today, even watch a day before you write your exam. You'll see that you will score high. All right. Fertilization means is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. That is the best definition. So when this fertilization occurs, it in outside is external. When it occurs inside, it is what? Internal. So take note of the examples which are supposed to be occurring in external fertilization and internal fertilization. So I'm going. You need to know the examples of each, because if you don't understand, then you won't be able to identify which organism belongs to which mechanism of fertilization. That's number one. Number two, oviparous, viviparous, and
recording in progress.
Oviparous, viviparous, and ovoviviparous. People confuse it with internal and external. So that's where another weakness comes from, because here the question can be which one is external, which one is internal, but which one is oviparous, which one is viviparous. Obviously, if it is vivi, there is no way how it's gonna be external. You understand? There is no way how. If it is ovo vivi, there is no way how it's gonna be external. Why? Because the development is inside the mother's body. You understand? So the mother is taking it as a shelter. So it is only oviparous, which can either be internal or external. So if you look at this is internal fertilization, yes, but still it is ovipary. This is external fertilization, but is ovipary. So it can be external oviparous or internal oviparous. So please don't confuse that, because students also confuse that. This is not too much marks, but I'm just trying to show you the small mistakes students you do in each topic, and then what you're supposed to emphasize on, so that you can get a distinction. Yes, so that one is easy. So we have seen that. just know the one advantage of external fertilization and internal fertilization. So basically it's the same thing here. Internal brings about internal development, and external still internal, there is high chance of fertilization. Internal development, there is high chance of the organism to survive. So yeah. This amniotic egg is rare to bring it like this. In most cases, we bring it in terminologies, and then we ask you about these terminologies. Yeah. Then you need to know precocia, atresia. Precocia, don't go to the paper without knowing precocia and atresia to differentiate the two. I said that pre means prepared. Precocia, prepared, born prepared. Eyes are open, you can see, you have down feathers, you see everything. Then atresia, all depends on the parent. Those are the techniques you can use to remember this so that you score a distinction. Yeah, so atresia, precocia, the difference you must know. So if you look at this, what is A and what is B? It is A, atresia, because the eyes are closed, it is check, it has no down feather, it can stand on its own. This one, eyes are open, it can stand on its own, has down feathers, you see. So please go when you know the two differences. If you have a question, let me know. Kind of missed your voice. Hey, how can you say that? You missed my voice. So I've spent time without talking about it. Can you please do male and female fertilization? I'm coming there. Don't worry, and I'm gonna tell you what you focus on more and the mistakes you do when I reach there, so that you don't lose that distinction. All right, let's continue to the next. So that is the topic. Parental care. Why do we need parental care? Basically, when we talk about parental care, is to bring about protection of these organisms so that they can reach maturity. So you prevent them from being attacked by the predators. So basically, that's parental care. I'm not teaching, just I'm trying to do the highlight. So that topic is done. You only require 15 minutes to read and read and read this, and then you score all the marks. Don't waste too much time on one topic, and that topic you just said, yeah, me, when you bring internal if you bring reproduction in vertebrates, I can kill it. You leave other topics, you go with the only reproduction in vertebrates. Hey, I'm not saying that you're gonna fail. So what I'm saying that don't spend like the whole almost half of the paper you're only means when you are revising this, and then spend also try to divide high. Let's go to the next topic, human reproduction. What are you supposed to focus on human reproduction? What are some of the things you can't skip when you go to human reproduction? Yeah, obviously the structure of the male and the female, you have to know that, and then the function of each. Yes, so I'm gonna just show you some of the few questions they can ask you when it reaches there. So labeling, you must know. Girls, I know you know this better than boys. Yes, you know that. Yes, so if they bring this, you there is no way you, a girl, to fail this. I don't know why you know it better than boys, but you'll tell us in the comment why do you know it better. Anyway, let's go to the functions of the reproductive structure. All right, I'm trying to check why is it behaving that way. All right, let's go to the next question. The next question is saying
recording in progress.
Sorry. All right. Then they're saying that what you need to look at, if you look at this question, all this is what I've talked about, the male and the female. Oh, anyway, if they bring this, it's a part of the male, you see. So we can bring this question in terms of sections. But what are some of the things which confuse students? Yes, number one, this epididymis. This epididymis. Yes, vas deferens. In most cases, when we bring it, student confused. They call it sperm duct. Please don't call it sperm duct. Call it vas deferens. It's better to call it vas deferens. So some of the things which you need, why do we have these testes hanging outside? Labeling, function of each part, please make sure you know. Why do we have these testes hanging outside? We say that they hang outside to provide favorable temperature for sperm production. Yes, sperms are produced 2 degrees less than the body temperature. Students make this mistake and they say that sperms are produced 2 degrees less than what? You have to be specific that less than body temperature. Or sperms are produced 35 degrees Celsius, at 35 degrees Celsius. So you have to be specific, straight to the point. Please don't leave my answers hanging, because we won't give you a tick if you don't complete your statement. Yes. Number 2, what if we cut this? What will happen? Will a person be able to spread HIV or STDs? What? Yes. Why? Because there is other fluids which are involved during this process of copulation, and they can be having some blood, so there is some contamination, and then HIV can be spread. If this vasectomy has been has been done on this person, can this person impregnate the person? No, because these sperms are no longer able to cross to the urethra. You understand? So the semen you produce is no longer what? Is no longer having sperms. Yes. What's the difference between semen and sperms? Semen is a combination of sperms and these fluids, while sperms is only this fluid which is coming, the one which contains the reproductive cells. All right, or the reproductive cells. Hey, how do you call these how do we call these glands? The 3 glands. These 3 glands, 1, 2, 3. We call them accessory glands. But sometimes when we bring it, people, they don't know which one is which. Okay, use this technique to know these glands. Number 1, when you start the testis, the first gland, you see this tube, you see this tube, you see this tube. The first gland you find, ne? Yes, this first gland is called seminal vesicle. So seminal vesicle is the first. The second one is prostate gland. The third one is Cowper's gland. You understand? So seminal vesicle, prostate gland, Cowper's gland. Please, if you are coming from the penis, the side of the zobza here, yes, the first gland is the Cowper's gland, the second gland is the prostate gland, and then the last gland is that's how you can identify it. So I think I'm done with that. Please don't make mistakes when it comes to that. Ne? Yes. Okay. Let's go to the next one. They're saying that I want code 7. Yes. You will get code 7. I'm telling you. You will get actually more than. Yes, you get it. All right. Sorry. All right, here you see now they're asking you some of the things. I'm not going to describe the process of spermatogenesis. Okay, I'll talk about it when I reach there. So when you try to answer this, you see during vasectomy, you see I talked about it. Explain one reason why men who does not have children choose to have vasectomy. You see, who doesn't want, meaning that you have cut the sperm from reaching the vagina. Yes, definitely. Why do testes hang outside? We have answered that question. Testosterone. Here, this is the hormone which is being produced from the testes. Testosterone has 2 functions. That is secondary sexual characteristics and then maturation of the sperms. All right. Now, when you go to here, you only need 2 things which you need to know here. The cells of Leydig, and the Sertoli cells. Cells of Leydig, what is the function of the cells of the Leydig? Is to produce testosterone. Simple. Testosterone is being produced from cells of Leydig testosterone is being produced by cells of Leydig. Oh, what's the function of the cells of Leydig? Is to produce testosterone. What about satellite cells? Satellite cells is responsible for nutrients. Done. Nothing else you need from here. Don't waste time. Don't waste time. Anyway, I'm not saying that don't read more, but don't waste time. Let's go to make sure that you maximize reproduction. Yes. Then when you come to female reproductive system, obviously you boys, you know this better. You know it better, actually. Yes. So if you know it better, there is no way how you're gonna bring it. But in most cases, when it comes to the male reproductive system, we bring this sideways. While when it comes to the female, we bring this way. You understand? So female, when they bring female, guys, it's always simple. But let me classify this or clarify this. This is a uterus. This is also a uterus. Sometimes I remember when they brought this and they asked you, what is that? And then someone couldn't answer that. I don't know that. This is also a uterus. He's asking you the head. The top part of the head, the sides of the head, the side or the lower part of the head. It's the same thing. It's the head. Yes. So endometrium is also part of uterus, but the uterus is divided into endometrium, the uterine wall, and the uterine cavity. You understand? Yes. So if the arrow is pointing here on the lining of the uterus, which you call the endometrium, then call it endometrium. When it is inside here, call it uterus. Or you can call it uterine cavity. It still is fine. When it's pointing here, call it uterus. If you call it uterine walls, it still is fine. What is the second name for the vagina? We said it is birth canal. Don't call this one oviduct. The same way we say you don't call it sperm duct. Call it fallopian tube. If they ask you my name, what's my name? Don't call me teacher. Call me M.SAIDI. Yes, that's my name. I'm not oviduct. My name is, you call it vas deferens. Its name is vas deferens. It's oviduct because of its function. It is the duct, the tube for the ovum. That's it. So know the functions, please. At least I've talked about that. Which hormone is responsible for secondary sexual characteristics? That is the estrogen. Yes. Please, I'm not going to talk about this too much. Another thing which students confuse is the difference between the vulva and the vagina. The vagina is the inside part, while the outside part, yes, is what called the vulva. Please don't call the vulva the vagina. Never. They're not the two. They're not the same. So vulva is the inside part. No. It's the outside part, what you see. While the vagina, yes, I think now you know. You're done with that. You require 1 hour to read. If you have read like this, and then now you require 1 hour to read human reproduction effectively. You require 15 minutes for reproduction in vertebrates. You see? So now keep calculating the time you will see. You see that you read the paper within 6 hours, you can get a distinction. If you have been reading, if you have not been reading, it's a different story. Okay, let's go to the next one. This is the ovary. What do you expect to, if they bring it, what do you expect, the questions they're going to ask you? Obviously, they're going to ask you, what is this? Is ovary. But don't call it, did I say ovary? Mature, call it Graafian follicle. But don't just call it Graafian follicle. This is immature Graafian follicle, while this is mature Graafian follicle. Why is it called mature Graafian follicle? Check. The ovum is there, it's big enough, and it's just next to ovulation. This is the process of ovulation. You understand? So this is the ovum. So this is the empty Graafian follicle, which turns into corpus luteum. So this is corpus luteum, you see, and then now, please go when you know this. You know, when you read this prediction and revision together, after reading it, when you go back to your book, life is gonna be easy for you. That's why I make it a little bit longer, so that when you watch it full, you get, you don't need too much time for you to read and read and read and read and read. Ne? Yes. This one is saying, just want to say I appreciate and wanted to ask how reliable this prediction. Reliability is 90% correct. Ne? So if you want a distinction, yeah, cool. You understand? Yeah, you read it, do what I'm saying, go to the paper. I'll do the discussion of the paper the same day. I'll do it live, and then we shall see. We shall talk and see who is right and who is wrong. Ne? Yeah. Anyway, let's continue. Let's go to the next sabu. I trust myself. Ne? I trust myself. Anyway, let's go to the next question. Did I complete here? Yes. So now, if they bring this diagram, look guys, another question here, cool. Another question here, cool. Ne? When they bring this, they ask you, did the fertilization take place? No. Why? Look at the corpus luteum. It's degenerating. It's degenerating. I said, watch this video today and watch this video a day before you write. Go write. We shall see. Ne? Yes. So, you see what it means? It shows that it is degenerating. Ne? So, it means that corpus luteum is degenerating. Therefore, fertilization did not take place because now this corpus luteum is going to produce less progesterone, and then once less progesterone is produced, then follicle stimulating hormone is going to be released. Done. Puberty in male, I am not going to explain that. What you need to know is which hormone is responsible for maturity, which is called testosterone. Please don't ask me that question. Does spelling matters? Definitely spelling matters. Please don't ask me that question because I won't answer it. Ne? Yes. Female reproduction, puberty is estrogen. It's being caused by estrogen. So, done. Characteristics, you know, I'm not going to waste. Here is the issue. Ne? This is the issue. Guys, formation of gametes, which you call the spermatogenesis and oogenesis. Ne? Yes. This formation of gametes. Ne? Spermatogenesis and oogenesis, I'm going to explain to you. Yeah. I know that there are so many books which have this complex process of spermatogenesis. Hey guys, don't waste time. I'm telling you, don't waste time. This is what we want. This is what we want. Formation, spermatogenesis, the formation of sperms. You tell us, cool, you get a mark. Under the influence of testosterone, you get a mark. The diploid cells in seminiferous tubes, you get a mark. Undergoes meiosis, you get a mark to form haploid sperm cells. A mark. Don't tell me other things. Don't tell me spermatids, primary oocyte, secondary oocyte. Don't tell me that we don't want those in spermatogenesis and oogenesis. Don't waste time. This is what we want. You understand? Yes, because when you are marking, we follow the examination guideline. Is it clear? Let's go to the next one. So, please, if you are to read the spermatogenesis, oogenesis, focus on that. If you don't have it, take a screenshot, then try to record it. Try to revise it. Structure of the sperms. What are they asking here? There are some 3-4 questions they ask here. You can't miss it if they bring it. All right. Naming is cool, you know it. Drawing, make sure that when you draw it, you draw with proportionality. Don't give me a very big head and a very small chest. Are you like that? No, man. Show proportionality so that we give you a mark. You understand? So head, acrosome, nucleus here. So where are students getting mistake? Here, they call this acrosome. When they ask you the functions, you tell me that acrosome contains enzymes which digest the membrane of the ovum during fertilization. Cool. The acrosome does not digest the membrane of the ovum, but the acrosome contains, listen carefully, it contains enzymes which digest. Because many students, they say that acrosome digests the membrane of the ovum during. No, you'll get a 0. I don't want that to say that statement that you go out of the paper, you think you have killed the paper when the paper has killed you. I don't want to say it, and I've not said it. So make sure that you do what I'm saying. Yes. We need more doctors, ne? Yeah, we need more doctors. Because for us, now we have grown up, we need people who are going to be replacing us. Yes, and it's you. All right, oogenesis. Let me tell you, how do they ask question you here. The first question, the adaptation of the sperms. The adaptation of the sperm for its function. Now you talk about long tail to provide propulsion force for forward movement. Cool. Long tail, propulsion force for forward movement. Don't just say for movement, for forward movement. New middle piece has mitochondrion, many mitochondrion to provide energy, the energy for sperm movement. Still, the head, uterus, the characteristic, the head has the nucleus for genetic material or inheritance. Acrosome contains enzymes which digest the membrane of you see, now those are adaptation full. But they can ask you a question, adaptation of the sperm for locomotion. Now you only talk about the tail and the middle piece. That's all. Adaptation of the sperm for fertilization. Now they are talking about only fertilization. You only talk about the acrosome and the head. Is it clear? Don't just go and then say, no, me, I know the sperm. Look at the question critically. You remember these words. All right, let's continue. Oogenesis. What are you talking about on oogenesis? Same story. Don't waste time with too much information in your books. End up cracking the head. We're not looking for that. This is what we are looking for. What are we looking for? Formation of the sperm, oogenesis, formation of the ovum, process under the influence of FSH, the diploid cell. Don't use FSH when it's terminologies. Yes, if it's terminologies, please don't use FSH. Say follicle stimulating hormone. But if it's not terminology, it's cool. The diploid cell in the ovary undergoes meiosis. Yes, mitosis. Yes, they undergo mitosis so that they form the numerous follicle. Only one cell inside the follicle enlarge. You understand? Which undergoes meiosis. You see? And then the 4 cells are produced, only one survives. The one which are produced, only one will survive. And how do we call that one which survives? We call it a haploid ovum. Done. Guys, we don't want these things. We don't want... we bring this diagram to know that, okay, this is 2n, 2n, this is 2n, this is now 1n, this is 1n. If this is human, how many chromosomes? This is 46, 46, 23, 23. You see, this, we bring this diagram just to confuse you with that. That's all. No complication here. No complication here, I repeat. Let's go to the next one. Ovum, this is simple. Very few things they ask you here. You understand? Yes. Labeling, cool, you know it. But sometimes students, they confuse. The outer layer is called the jelly-like layer. The next layer you see here, we call it what? This one, cell membrane. This one, cell membrane. I think I didn't label it. This one, all the cells, you call it cell membrane. The inside one is what you call it, what we call the cytoplasm. Then the dense body is what called the nucleus. That's all. Whenever they ask you the adaptation of a nucleus, think about genetics, inheritance. That's it. Is it clear? Guys, let me know if you are in class. Let me see who is saying. They're saying that I just wanted to say okay. They're saying that thank you so much. You're welcome. Who is this? Amanda. Okay, Amanda. All right, let's continue. Guys, I repeat, don't make a mistake not to watch this prediction 2 times before you go for the paper. Is it clear? Because it's going to help you to read and also help you to find what I said in the paper. Let's continue. Can't electricity is coming back from where it's produced from. Anyway, we shall find a way out. All right, menstrual cycle. This is a problem for students. Let me just show you what you do when it comes to menstrual cycle so that you don't stress. Yeah. All right. They're saying menstrual cycle. Menstrual cycle, what do you look at menstrual cycle? Menstrual cycle is divided into 2, the uterine and ovarian. Uterine is dealing with this uterus. Ovarian is dealing with the ovary. We talked about the ovarian already when I was talking about this. Yeah, so I explained that. So it's not a big deal now. So please make sure. You see, I talked about this. So guys, make sure that you know. So, but the uterine, you need to know the hormones which are produced here and the function of the hormone. That's all. The uterine is done. So, did I say uterine? The ovarian is done. Hormone produced and the function of that hormone. That's all. Uterine also is the same story. Here you see now you have this cool. You have 4 hormones: follicle-stimulating hormone, luteinizing hormone, estrogen, and then progesterone. These two is they are produced in the ovary, but they work in the uterus. Therefore, they are produced from the ovarian cycle, but they work in the uterine cycle. These two, they are produced from the brain, but they work in the ovarian cycle. So just know these 4 hormones, their function done. Where do they work? This thing of menstrual cycle is done, man. It's simple. Make things happen. Don't let the things to make don't make the reverse. Anyway, okay. Look. Here, this one is developing. So it's under the influence of, remember, these 2 hormones. When you're talking about the development, you only talk about these 2 hormones which come from the brain. Follicle-stimulating hormone. This one is developing because of the follicle-stimulating hormone. Follicle-stimulating hormone. A hormone which stimulates the development of the follicle. Simple. Done. So, it is follicle-stimulating hormone. Follicle-stimulating hormone, which helps there. And then now, the second hormone is luteinizing hormone, which triggers this to happen. So, ovulation is under the influence of luteinizing hormone. Done. But now, and then number 2, it also helps to change the empty Graafian follicle to corpus luteum. It stimulates, guys, luteinizing hormone does not change. It stimulates the conversion of the empty Graafian follicle to corpus luteum. It stimulates. We need to hear that word, stimulation. All right. Now, when this is developing, what's happening to it? It produces a hormone called estrogen. Done. We don't want to know the function of estrogen here. We shall see it. We shall know it in ovarian, in the uterine cycle. Then this one, which hormone does it produce? It produces progesterone. Done. Uterine cycle is done. If you read here, is what I've said. Development of the Graafian follicle under the influence of luteinizing follicle-stimulating hormone. You see, ovulation occurs, and then the empty Graafian follicle changes to corpus luteum. Done. You see. Don't stress yourself, guys, man. Don't stress yourself. Uterine cycle. Now the hormones we produced here, they come and work here. Estrogen stimulates the endometrium, become thicker, vesicular, and glandular. Thick, vesicular, more blood vessel, glandular, more glands. Simple. And then progesterone. Remember we said that it produced from corpus luteum, make the endometrium more thicker, more vesicular, and more. We want to see this word more. You understand? More thicker, more vesicular, and more glandular. That's it. The uterine cycle is done. You understand? If the level of progesterone drops, check. You see, menstruation is going to happen. The lining of the endometrium, the lining of the uterus, that is the endometrium, will break down, and then this is called the menstruation. You will see the nice blood. Yes, some ladies are saying that, but it's not nice. So if it's not nice, are you nice? It's nice. I mean, I'm saying it's nice. Yeah, it's nice. So anyway, doesn't mean because it's smelling bad is not nice. Or whatever is smelling. Let me just leave it. There are so many things which does not smell good, but they taste nice. An example is the egg when you are eating it. The neighbor, what is he saying? But who is eating? What are you saying? Anyway, that's not a prediction. Yeah. Guys, leave my ladies. So please, when that happens, please help them. Yes, help them. This is the menstrual cycle. The hormonal control of the menstrual cycle. Do I just here I've talked about the how to interpret it. So here what you need to focus on here is number 1, the function of this, the function of this, the function of this, the function of this. I'm not going to explain this, you understand, but you know, if you draw this, this works here, this works here, this works here, this works here. So they cannot ask you anything here in this region and you talk about progesterone, in this region and you talk about this. Never. Okay, now they ask you, did the fertilization take place? What are you looking at when they ask you, did the fertilization take place? Look at the level of progesterone. If it drops, no. Look at this, the endometrium. If it shrinks, menstruation is there? No. Look at the corpus luteum. If it becomes smaller, then fertilization did not take place. Please know that. Which hormone is highest at day 14? Luteinizing hormone. Which indication shows that ovulation occurred on day 14? There is a release of ovum and there is an increase in the level of luteinizing hormone. You see? Yeah, why? Because luteinizing hormone is triggering ovulation. So this question is, in most cases, when they bring it, sometimes students go off. But don't stress, relax, do other questions, come back to this, you'll be able to answer it. If you see that you don't understand it, yes. Okay, I'm gonna show you some questions so that maybe we rush. Yeah. Negative feedback mechanism. What happens if progesterone levels drop? What is the influence of the decrease in the progesterone on follicle-stimulating hormone? You see? So they can ask you that question. Simple, they give you 3 marks. So definitely, if progesterone levels decrease, then follicle-stimulating hormone is gonna increase. Why? Because now the brain is gonna be stimulated, showing that, oh, the person is not pregnant. Therefore, you're supposed to prepare this person again for pregnancy. That's it. That's why if you're pregnant, we don't check anything. This pregnancy test is checking the amount of progesterone level in the blood. That's it. Okay, copulation. Copulation, obviously, is sex. But you explain it. So, I'm not going to. Fertilization, I said, when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form the diploid zygote. That's the best answer. If you have another answer, cool, but make sure that you write what they gave you in the booklet. Yes. Process of fertilization. Hey, here is simple, but sometimes when they ask you this, I'm gonna show you the question that we're gonna bring. They might ask you, and then you fail. You bring the process of fertilization. Haploid sperm cells. Remember, they have the enzymes acrosome, which digest the membrane of the ovum, and then the nucleus of the sperm enters and it fuses with the nucleus of the ovum, and then the diploid zygote is formed. These blue color things are the marking points, what you are looking at. If you don't see them, 0. So please, I don't want you to get a 0. I don't want it. Hey, funny time, man. How can you say that? How can you say that? Hey, it is not nice. You are nice. Hey, I was just asking. Guys. Okay, let's continue, guys. Hey, let's continue, guys, man. You must ask your friend. If they say it's not, then yeah. Anyway, so fertilization is cool now, guys. Make sure that you you get it, You get it, Yeah, I mean you get it. I'm not the one who said that. Hey, this one. This is where the problem comes from. When this bring here 6 marks and then you see someone is failing to get it. Man. When they ask you, explain what happens after fertilization, please don't include the process of fertilization. This is what I was saying. Now talk about the sperm comes and then the enzyme on the sperm. They didn't ask you that. They said explain the development of the zygote until implantation. Don't talk about fertilization. Because now, the process of fertilization, how does it occur? You're wasting time, man. Yes. So, the sperm, the ovum, the sperm. I see. By the way, next year, I'm going to be full here to help anyone. If you have a sister in grade 11, grade 10, make sure that they subscribe so that they get full attention from this channel. Thundereduc and M.SAIDI. Alright. Here they're saying that, please don't talk about you tell me stories that now the acrosome, you told about the enzymes. No, they didn't ask you that. Right. What happens after fertilization? Immediately after fertilization. So you tell us a zygote is formed. After fertilization, a zygote is formed, which undergoes mitosis. We give you a mark, we give you a mark. To form what? It undergoes mitosis. You see this? I remember when they brought this question and then they ask you what is this? Because you see it's one cell. Say someone, what is this? Is what? Yes. Sometimes we make them like this. 1, 2, 3, 4, 5. So those are 5 cells. It's not yet a morula. A morula is many cells you can't count. You see? Yes, because even the others that side. Yes. So this is 1, 2, 3, 4. If there are 2, just say it's 2 cells. You understand? So here it is a zygote. Yes, it is diploid. You understand? It's diploid. This is haploid. This is also haploid. Form a diploid, form morula. So after fertilization, a zygote is formed, which undergoes mitosis to form a ball of cell. This is a mark. Don't say to form morula. Form ball of is a mark. Called morula is another mark, which divide mitotically or repeatedly to form halo. This is also another mark. How do we call this halo ball of cells? Blastula. You see that is a halo. It has a hole inside. Hole. Ne? Unfortunately, you don't see my face. But a hole. Ne? Yes. So think about a hole. That's it. It's a blastula. So that's how you can remember. If you don't know a hole, ask your neighbor. Yes. Anyway, to form outer layer called chorion. Chorion. The outer layer. This outer layer is called chorion. You remember, sometimes they ask you that the outer layer of the baby, how do you call it? It's chorion. Because they said it here. It's called chorion, which forms finger-like projection. This is when you start vomiting because something is itching you down there. You start vomiting and say, hey, what's happening? You start to sleep, to doze and everywhere. Yes, something is happening inside your body. Finger. Imagine someone is... let me just keep quiet. I might be abusing my... Okay. But finger, like, fingers. When someone, for example, in the stomach, brings a fist, and then you stress the finger. How do you feel? You feel the pain. That's exact. Finger-like projection. How do you call those finger-like projection? Chorionic villi. What is it for? Implantation. Simple. Guys, hey, don't go to the paper. I'm saying this as if it is a secret. Don't go to the paper when you don't know this. Yeah. Please make sure I know it. Anyway, let's go to the next one. So now if they ask you this, yes, this is what I was saying. What is this? This is a zygote. Cool. Fertilized ovum, fertilized egg. So it's a zygote. So what is this? It's 2 cells. What is this? It's 4 cells. What is this? It's 8 cells. But nowadays, you can't count them. Therefore, it becomes morula. And then this, which has the hole inside, we call it a blastula. And then implantation has taken place. That's it. Life science is good. Here are some of the questions. Yeah. So please don't make a mistake to go when you don't know that. Yes. Implantation. Yeah, cool. Can bring it in the terminology. Yeah. This is if you are struggling with this life science, not that big deal. Chorionic villi, amniotic fluid. Please go when you know the function of the amniotic fluid. Functions of amniotic prevents dehydration, keeps temperature, and then allows movement of the baby. Yeah, guys, there is this thing whereby students, they say that amniotic fluid is responsible for nutrients. Guys, you will get a big 0 with the eyes. Stop saying that. I don't know where you got it from, because no one has ever said that in any book. You are just speculating. Please, amniotic fluid is not responsible for nutrients. You understand? Amniotic fluid is responsible for mechanical shock or shock absorber, or cushion against mechanical injuries, prevention of dehydration, keeps the temperature almost constant, not constant, almost constant, and then allows free movement of the baby. You understand? Please. Umbilical cord. Yes, they brought this and then students, they fail. Fail, please. Artery brings blood to the heart. Artery takes blood away from the heart, while vein brings blood to the heart. So this is the heart. of the baby. So we are referring to the heart of the baby. So the blood vessel which brings blood to the heart is a vein. And because it's coming from the mother, so it will be rich in oxygen and nutrients. Do you understand, guys? And then the blood vessel which goes away from the heart is called the artery. Because this artery is going away, it goes to the mother, therefore it have more carbon dioxide and waste product, meaning that it will have deoxygenated blood. That's the trick behind, nothing else. Here, done. This is the, how do you call this? This is a placenta, this is umbilical cord, this is the fetus, this is the mother's. Done. Guys, man. Okay, no two functions of placenta. No two functions of placenta. Nutrition, protection, gaseous exchange, excretion. If you don't remember the functions of placenta, okay guys, this is what you do. Think about the processes which occur in your body. Obviously, you have gaseous exchange, you excrete, you go to pee, you develop the boobs, that is endocrine, then you protect, the skin protects you, you eat, and then you fart, man. Nutrition, yes. So think about the processes you have in your body, then you'll be able to know the functions of the placenta, because the placenta is doing everything for the baby. At least not two. Don't know everything, man. Two, yeah, okay. That is, you require 1 full hour for you to discuss about this, for you to read reproduction. Let me show you now questions. I'm not going to waste too much time. I'm just going to go highlight. Yes. So we talked about this. Identify part process involving in process that lead to formation of the ovum or genesis. We talked about it. So you see, fertilization. What is this? Is the nucleus. What is this? What is this? Is a cell membrane. What is this? Gel-like layer. So you see, now you know. Please make sure that you know how to label these things. You see, now this process, obviously, you know, this is, what is this? It's a ball of cells, which is called morula. And then what is this? You see, as if there are two nuclei. Check. There is this, there is this, there is this, and there is this. So definitely, this can't be ovum. So this is a zygote. And then because it has a hole, so it becomes a blastola. You see, things are simple. Simple things. Guys, make sure that female, I told you guys, there's no doubt that you know this. What is this? This is the cervix. This is not the vagina. Vagina is this big one, which can, by the way, it can be, it is elastic 200%, 200 times. Yes, it can become elastic. Even if you're as big as you can't, it will accommodate you unless you are an elephant. Anyway, I'm not saying that. I'm not the one. Don't say that I said it. Anyway, I'm a lifetime teacher. I'm just teaching. So you see, this is Zobza. Make sure that you know this Zobza very well. Ne? Please go when you know this Zobza. Ne? This male reproduct. Don't write Zobza in the exam. Ne? You'll get a 0. You'll get a 0 with a beard. Okay. Name the part which is surgically cut during sterilization. We say that is this vas deferens B, which the process called vasectomy. So I'm just choosing a question to see if we can answer it. Ne? Okay, let's just choose one. Okay, this. Where is fertilization occur? In the fallopian tube. You see? It occurs in the fallopian tube. Not oviduct. I said it. Simple things, ne? Yes. So make sure, guys, you know. Explain why is it necessary for D to be outside the body of the male. We explain it. We say that to provide favorable temperature for sperm production 2 degrees less than the body temperature. That's it. You see? What is H? Now, this is the vagina. But if it was here, it would have been a vulva. You see? So, guys, you can do it. You see this? What I told you, that when they bring this diagram, they're asking you how many chromosomes. They're not asking you these oocytes, spermatides. No. No one is going to ask you that. Yes. So, spermatozoa or mature ovum. So, you see this? It's simple. We say that. Check now. They have reversed it. When I was explaining it was like this. So, they have reversed it. So, guys, you will answer these questions. Yeah, last time when they brought this, people failed. So this is the ovum, but this is fertilization. This is a zygote. These are 2 cells. You see, these are 4 cells, while this is a ball of cells, which called more ruler. So yes, so this amniotic, you see, we also talked about this placenta. This is the outer layer, this one, ne? Yes, which called the chorion. The outer layer, how do you call that outer layer? We call it the chorion. So guys, I know you're gonna get a distinction when it goes to that. Did the fertilization take place? Check. Yes, took place. It did not take place. Yes, because progesterone decreased. And then check the size of corpuscular degenerates. Therefore, fertilization did not take place. What indication show that ovulation occurs on day 14? Because of this ovum is released and then the level of luteinizing hormone is high. So, hi guys, you will get a distinction, you want it or not, as long as you follow what I'm telling you. Yes. Anyway, let's go to the next sub topic, which is the nervous system. Nervous system. He's fast. He's fast. Serious? Am I fast? It is not nice. Big zero. Oh man, sorry. Anyway, I was just reading the comment you were saying. Guys, I'm coming just in 1 minute. Just in 1 minute. Just in 1 minute. So where we are going to go to this and then just show you what you have to talk about, man. What you need to talk about. I'm just scrolling with the distinction material just to reach here. Just to reach here. Yes. I'm coming back, guys. Just 1 simple minute. Yeah, I'm back, ne? I'm back. All right. Yeah, let's go to the next topic. That is nervous system. Oh, basically, let me check something. Yeah, if you don't hear me, you let me know. All right. Nervous system, what you need to talk about here is simple. Know what is a stimulus because it's going to confuse you. If you don't know what's a stimulus, impulse. Ne? Yes. Okay, here, if they bring this, central nervous system, brain, spinal cord, center, spinal cord. Just check your head, the way you go down until the tail. I know you have a tail. If you don't have a tail, when you're bathing, go check yourself. You'll find that you have a tail. I'm not talking about only boys, even girls. I'm not talking about, you can't have a tail in front winner. I'm talking about the tail, yes. So you have peripheral means sides. So you have autonomic nervous system and somatic. Autonomic nervous system, you have the sympathetic and the parasympathetic. Sympathetic, it prepares you for emergency. Parasympathetic, it brings back to the situation back to normal. Sympathetic work together with the adrenaline. Yes. So basically, that's what you need to know there. Know if they just bring the labeling. How is the brain protected? Cranium, meninges, ne? And then cerebrospinal fluid. Don't call it. There's a difference between a cranium and the skull. Ne? Yes. So cranium is better to say cranium and meninges. You don't want to know the names. However much people are talking about the names in the books and stuff, don't want to know. Yes. And how is the spinal cord protected? It's protected by the cerebrospinal fluid and then the vertebra, yeah, or vertebrae. Function of the brain, obviously. Everyone knows the brain, eh? Please, unless you don't have a brain. Everyone has a brain, so how can you feel something you have? Okay, function of the brain, simple. At least know two of each. Yes. So, memory, reason, judgment, that is higher thought process. It's the biggest part of the brain. Which part connects to left and the right hemisphere? That is corpus callosum. And then they're asking you, there's something whereby students get fail. Here, the first swelling is not medulla oblongata. It's called the pons, but we don't ask you it. Then the second swelling is the medulla oblongata. After that swelling down is the spinal cord. Please don't confuse medulla oblongata and the spinal cord. Ne? Yes. Voluntary, this one, coordinates, this one, coordinates all voluntary movements. This does not control voluntary actions, voluntary movement, and does not control voluntary movements, but it coordinates voluntary movements. Control balance. This is balance, muscle tone. Cool. You understand? What controls voluntary actions is this biggest part of the brain. So I think now you are getting it there. Ne? Medulla oblongata is this one because it is next to the spinal cord, and the spinal cord is for involuntary actions. So you think about involuntary actions. Yeah. So here we are talking about breathing, heartbeat. However much you say that someone is controlling your heart, not in science. Maybe that is in that other world. Yes. Anyway, so please make sure that you know how to label the brain and these functions we have talked about. So brain is done. So you don't having too much in there. Spinal cord. Sympathetic and parasympathetic, at least you know how including it with adrenaline. Eh? Yes. So let's go to the axon. Dendrite, cerebellary, axon, then myelin sheath. Dendrite transmits impulse to a cerebellary. Cerebellary, that's where activities take place. Then axon transmits impulse away from the cerebellary. And then myelin sheath, that is a sheath. When you talk about the sheath, is a protector. I used to tell students that it acts like a condom. Guys, stay virgin. Eh? If you lost the virginity, start now. I'm saying that sheath is a if you look at the wire, it's naked. Eh? It's naked. If you touch it, the electricity will shock you. But now, if you're using a sheath, it is easy. It is easy to touch the wire because it is protected. So basically, sheath is for protection, insulation. All right. So motor neuron, cool. Difference between sensory neuron and motor neuron. In most cases, when you ask you the differences, you must know that. Two, at least go when you know two. When you know how to draw this and the sensory neuron, no one is gonna bring the connector. No one. It's this and this. That's it. And they have the same parts. How do I differentiate between this one? The cell body is along the neuron, while this one is at the beginning or the terminal of the neuron. Simple. Reflex arc. We have 5 questions here we ask you here. What is the reflex action? What's the reflex arc? Function of the reflex action. What happens if this is damaged? And then what happens if the other one is damaged? Simple. And then reflex arc is done. Reflex arc is a pathway along which impulse is transmitted. To bring about a response. That's a reflex arc. Or you can say that a reflex arc is a path traveled by the impulse during the reflex action. Simple. Path traveled by the impulse during the reflex action. Simple. Reflex action, you talk about rapid automatic response to the stimulus. Simple. Sometimes you say that rapid involuntary response to the stimulus is still simple. You understand? Yes. So please know that. What happens if the motor neuron is damaged? You won't be able to feel, but you will be able to respond. I gave you examples there. What happens if motor neuron is damaged? You will be able to feel, but you won't be able to respond or to react. Simple. So, examples of reflex actions, you talk about blinking of the eyes. Yeah. So, guys, go when you know this by heart. If they ask you the transmission of the impulse, you just start receptor, tell us the function, receives the impulse, and then interprets it, then talk about the sensory neuron, tell us the function, transmit the impulse, talk about the connector, function, motor neuron, function. That's it. Just part, function in the order. How do you know that this is sensory neuron? Because sometimes they bring it upside down. Just look at where the swelling is. That neuron is going to become the sensory neuron. Why? Because the sensory neuron has the cell body along it. So if you look at it here, yes, the cell body is here. So then the neuron continues. So if they bring it upside down, whatever they have done it, so that they can confuse you, make you fail. Tell them that you found a captain, not just this one who are just starting. So that's done. Functioning is the same thing. Part function. Significance of reflex action, we say that protects the body from further harm. Yes, you protect the body from further harm. Examples, knee jerk. Please know that. Examples of reflex action. Nothing too much to read here. Synapse, what you need to know here is a junction between two or more neuron and what its function. That's it. No one is going to ask you how to it operates. No, man. You're not at the university or the college, no. Function of the synapse, it allows impulse to move in one direction. Simple. Allows impulse to move in one direction. I repeat, allows impulse to move in one direction. Ensures impulse to move in one direction, prevents continuous stimulation of neurons. Yes. These are some of the questions. I'm not going to go through them, but you will be able to answer. Alzheimer's disease, we like it, and multiple sclerosis in objectives. So Alzheimer's disease, you have to know what is destroyed. Basically, is the brain, and then the multiple sclerosis, what is destroyed is the degeneration of the myelin sheath. Sometimes you call it the degenerative disease. Done. Then let's go to the receptors, eye and ear. Here is simple thing. Eye, ear is a simple thing, guys. What do we talk about here? Function part. Let's not talk about this too much, but make sure that you know function part. Yes. Okay, before I go there, let me just show you here. Before I go to the eye. Saying, sir, don't forget geography. We are writing on Tuesday. Geography, you are writing on Tuesday. Yes, I know. Don't worry. We're gonna fix you. Guys, this channel is here for you. Is here for everyone, every student in Mozambique, so that we see that, and it will bring you the best teachers. Yeah. If you see that this teacher is not, let me know. Let us know. No, I will bring, and you know, or you have someone you to recommend. Tell me, or tell us. We will bring that person to board. Yeah, we'll bring that person so that you are helped out. You understand? Yes. Anyway, let's continue. Let's continue, guys. So guys, so brain, we talked about it. You see the nerves, we talked about that. Brain, we talked about it. And guys, if you see any question here, please let me know. You see label, all this, guys, you're gonna pass this. You're gonna pass this. I think I've tried to emphasize what happens if this is damaged, what happens if this is not working. Guys, I'm saying, okay, the ear. Let's go back to the eye. What are you supposed to focus here? Look at the pupillary mechanism. Pupillary, guys, students confuse this, and I don't know why. Pupillary mechanism is the ability to see in dim and bright light. You understand? Accommodation is the ability to see near and far object, meaning the lens is shape changing the lens. While binocular is the ability to see with the two eyes. So I think now I'm clear. So this one is to see eyes, to see with the two eyes. Accommodation, no, at least these definitions. Better to see, or they change the length of the focal length of the lens to see the near, or to focus on near and the distant object. You have to know that. Yes. Near object, far object. Guys, don't memorize all. Memorize near, it will be the reverse for the far. You understand? Near object, guys, whenever I talk about accommodation, when they say near, from far to near, explain what they have said last. From near to far, you explain far. From far to near, explain near. You understand? So now, when you're explaining, please don't talk about the pupil. Don't talk about the iris, sacral muscles, radio muscles. Please don't talk about that. Here, you only talk about the what? The ciliary muscles, and then the suspensor ligament, and then the lens. Simple. Ciliary muscles contract, suspensor ligament slacken. Slacken means they relax. Don't say relax, say slacken. The tension of the lens decrease, then the lens becomes more convex. It becomes more thick, and then light rays are reflected or bent more, and then clear image is focused on the what? On the retina. Guys, these two, they work antagonistically. Yes. If ciliary muscles contract, suspensor ligament relax. You see them that they are relaxed. You see them that they are what? Now this thing is jamming. Why is it jamming? Taking my time. Anyway, it must not jam. Not jam, ne? It must not jam. Guys, if you have questions, let me know, ne? I'm here for you. I'm here for you. For you. Yeah. Okay, it's back. I'm saying, you see that they are relaxed. You see? So if this relax, this contract. If this, we call it become taut. If this contract, this one relaxes. You understand? And how do I know that it's near object? When they talk about more than the 6 meters is far object. Less than 6 meters is. How do you see a near object? You see it big. Therefore, the lens is supposed to be big. How do you say far object? You see it's too small. Therefore, the lens is supposed to be small. How do I make this lens to be big, from small to big? It means that the tension must be relaxed. If the tension is relaxed, it means that the ciliary muscles must do what? Contract. Then the suspensor ligament must slacken. You understand? And then now this lens, the tension is going to be decreasing, and the lens is going to become more thick. You understand? Which you call the more convex. If you don't remember the word more convex, you can say more thick. We also give you a tick on that. So this is the difference. So make sure that you memorize one. Don't go with too much information. Like, if things are not going well for you, man, don't stress. And the more you panic, the more things go out of their body. So what you need to do is you relax. Don't eat too much towards the exam. Give at least 30 minutes after you have eaten. You understand? Yes. Relax. When you relax, things will come out. You'll be flowing. You'll be remembering me, ne? Yeah, you just write because I said this, ne? That's why sometimes I don't just talk, and I go. So that I'm just highlighting. People like mechanism. We have the bright light and dim light. What do you... In bright light, you don't mix things up. While in dim light, you mix things up. So R with R, C with C. So R, radio muscles relax, sacral muscles contract. You don't mix things in bright light. You understand? Yeah, that's why some people, they switch off their lights at night. Which people are those? I don't know. Why? Because they want to mix. They want to mix what? I don't know. They want to mix R with the C, C with the R. Meaning that in dim light, people are mixing things up. R, radio muscles contract, sacral muscles relax. The diameter of the pupil, becomes big, ne? Yes, the diameter becomes big. And then what happens? More light will enter the eye. Ne? Yeah, that's dim light. While bright light, people don't mix. They're scared to be seen. R with R, C with C. Radio muscles relax, sacral muscles contract. And then, if what happened, less light will enter the eye. And then what happens? You'll be able to see in bright light. So basically, that's it. So please make sure that you memorize one. Bright light, R with the R, C with the C. You don't mix things up. While in dim light, you mix them up. Done. Don't waste too much time, guys. Yes. This one sometimes is a challenge for some people, but it's not that a big deal still. Yeah, it's not that a big deal. So let's see what it means here. Eye defects. Eye defects, short-sightedness. It means when you say short sight, it means that you can see things which are short, near. Long sight, it means that you can see things which are far. And then astigmatism, it means that you can see things. You can see things, but you have some problems with the edges. Then cataracts, the lens has clouded. It means that it cannot go through, so you'll be seeing things clearly. All right, okay, let's go back here. So please know that when you say short sight, what causes the short sight? You understand? And then what is the remedy for that? Short sight, what causes it? And then what is the remedy for that? You see? And then long sight, you see things which are far, but you can't see things which are near. It means that the object is forming behind, you see, behind. So what do? I need to bring it back, so I put a convex there. But short sight, it means that things are formed near here. So what do? I need to diverge it, you see, so that now this light appears to be as if it's coming from a near object with a big beam. Yes. And then astigmatism, this one, you have to put on glasses which are prescribed by the doctor, and you can also do the laser therapy. You go for real surgery. You know, if you watched Final Destination, that one was doing the laser therapy, and then Final Destination came. Anyway, treatment, cataracts, cloudy, and then treatment, surgery, or you replace it with a synthetic artificial lens, the lens which has been synthesized from the lab or from the what? From the factory. You're gonna kill it. Maybe the diagram here. Which part is yellow spot? Yellow spot is the part where they have the sharpest image which is formed. Yeah. Now I'm talking about the questions. Sharpest image is formed. Why do we have the sharpest image? Because we have the highest concentration of photoreceptors. That is the rods and the cones. Yes, the rods and cones. Why do we have no image when a blind spot is because the image is because there are no photoreceptors there. And then functions, other parts, I'm just talking about few things. And then this is the iris, and then the space is the pupil. You understand? The space is what? The pupil. So basically, the iris controls the space of the space, which called the pupil. So sometimes people confuse the iris with the pupil. And then this is a lens. What's the function of a lens? Is to bend light to the retina. So basically, it bends, refract light to the what? To the retina. So I think I'm clear there. Ear is, guys, ear is not that a big deal. We can just label functions, but let me show you where students go off when it comes to the ear. Someone saying what? Grace, I don't understand what you are saying. Mashallah, Grace. Anyway, if you have a question, guys, let me know. Let me know. All right, the ear. Pinna collect sound and directs in the auditory canal. Guys, ear is simple, ne? Please use Miss, ne? Use Miss, Marias, Incas, and then steps. It's better that way. It's the same thing, oviduct, sperm duct. This is English, this is science. Yes. So here, guys, know the parts and function, eh? Know the parts and function. But these two functions, three functions, make sure that you go when you know them very well. Eh? The first one is round window. The second one is eustachian tube. The third one is, who can guess the third one? So make sure that you know the round window, you know the eustachian tube. Eh? Yes. And then this part, the cochlea and the semicircular canal. So round window, it absorbs or releases. It's the same thing. Absorbs excess pressure from the inner ear or releases. When you absorb, you release what you have absorbed from. Release the excess pressure from the inner ear. And then over window, what does it mean? Over window. So basically on over window, what do you need to talk about? This is just create pressure wave in the inner ear. I didn't talk about that too much, but eustachian tube, you need to know, equalizes pressure on the either side of the tympanic membrane. How does it equalize pressure on the either side of tympanic membrane? Yes, how does it? Yes, equalize pressure on the either side of the tympanic membrane. Basically, this eustachian tube, it allows air saturation to go through in and out, so that now the pressure is what? Is equalized. Yes, they brought this question, I don't remember, I think this year, I think, and then students were off. Yeah, it equalizes pressure on either side of the tympanic. Actually, they asked tympanic, how does the eustachian tube equalizes pressure on the either side of the tympanic membrane? And then many students, they knew that it equalizes pressure on the either side of the tympanic membrane. But how does it do it? By allowing air saturation in and out of the middle ear through the tympanum. Simple. How do you call this? Oscos function, amplify the vibration. Also transmit sound, but amplification of the vibration, no it, ne? Yes. Which sensory cells which are found in the cochlea is organ of Cort, and which sensory cells which are found in the semicircular canal is the maculae and the utriculus. Simple, you are done. Hearing process, name, part, function, and in their order. Then the hearing process is done. Balance, 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 balance. You can use SAIDI's mode of balance. Change in the speed and direction stimulates the maculae. Change in the speed and direction stimulates the cristae found in the ampulla of the semicircular canal. It's a mark, a mark, a mark. Change in the position of the head stimulates the maculae found in the circulus and the utriculus. A mark and a mark and a mark. You see? The stimulus, now these are stimulus, ne? Yes. A converted stimuli are converted into a nerve impulse. Yes, is another mark, which is transmitted along the auditory nerves to cerebellum for interpretation. Simple. Done. Hearing done. So I did this model of balance just to simplify work for you. That's why it's called SAIDI's model of balance. Yes. So guys, that's it. Balance is done. Yeah, you guys are gonna kill this paper, I'm telling you. Ne? Yeah, practice more questions. Go to Thundereduc or Get Answered, one of the websites. You'll be able to get questions there and then answer. Try to pass, guys. You want you in the university. Yeah. Middle ear infection. Middle ear infection means there is excess fluid which has been accumulated in the middle ear and is caused by a bacteria. Treatment, the bacteria is antibiotics. Simple. Then number 2, insertion of the grommets. This is what you call the grommet. It allows air saturation and then it tries to drain the pus. I don't know how you call it. You call it pus. I don't know why you call it pus when it starts with U. I don't know. If you start it with U, then it's up to you. I've called it pus. All right. Deafness. You know that some people are deaf, but we have partial deaf and the total deaf. So when it's total deaf, then we can talk about these implants, hearing aids, cochlear implants. If it's partial deaf, in most cases, it's caused by accumulation of the wax in here, which sometimes we call it cerumen. How do you treat it? By removing the wax. Simple. Topic is done. Tell me, what is difficult there? What is gonna make you fail on that, on what we have talked about now? Tell me. There's nothing. I'm telling you, there is nothing. And you're gonna get a distinction. You're gonna get a what? A distinction. Let's wind up with these few small topics and then, yeah. Revision here. There are so many questions you guys, you can practice them. Then you won't even regret at a single moment. Hey, here is simple, né? Let me just wind up in very few minutes. So now, guys, you have 100 marks. Yes, out of 150 at least. Eh? Let me check. Yeah, you have 110, something like that. Yes. So here, endocrine, exocrine. Which gland is both endocrine and exocrine? Please don't forget about it. What's a hormone? Don't forget about it. Eh? Yeah. So this is look. Hypothalamus, pituitary gland, adrenal gland, thyroid gland. This is the one which is both endocrine and exocrine. Eh? Please know that. Know the part and the function. If it is making you a big deal, at least scope. To get a distinction, you don't need 150 out of 150. You need one, at least to be on the safe side, 125. See? Yeah, 125. Kill all the objectives, kill all the terminologies, kill all. You're gonna get it, man. Then we talk. So, guys, I'm not going to go through this. If you have any challenge, there is too much videos I've created on this and it's explaining you very well. Yes, but this is what I need to talk about. Glucagon and the insulin, they don't convert glucose to glycogen, but they stimulate. This word is stimulation. They don't convert, they stimulate the conversion. So you guys learn how to use these words. Stimulation. They stimulate, but they don't convert. Sorry, man. Okay. Negative feedback mechanism. Here, I'm just going to give you one trick how. But don't make a mistake to go when you don't know the thyroxine. Should I say it? Let me just keep quiet. Okay. Thyroxine, make sure you know it. Hyperthyroidism, hypothyroidism. If you swelling of the thyroid gland is called goiter. This one, goiter. But I'm gonna show you how you can answer questions concerning about homeostasis and negative feedback mechanism. Okay, you start with this. We have a first box which is normal level, whatever you want to talk about. Normal level of maybe salt in the body. Then you talk about if there is decrease or there is increase. If there is decrease, the last statement here must be increase. whatever you are increasing. If there is drop here, the last statement here, it must be increase. It's the same thing. If there is increase here, the last statements must be decrease. So you have 2 points to fill in. So you draw that, and then now you fill in. In most cases, they don't bring 2 of them. They just want 1 cycle. So now if it is salt, you know the which gland and which hormone is responsible for that. So if the salt level have decreased, the last statement must the salt level increases. So which gland is which hormone is responsible is aldosterone, and which gland produces aldosterone is adrenal gland. So I'm gonna come here. The first one is the gland producing the hormone, and the second one is the function of the hormone. Then they will link. So aldosterone, so not adrenal gland will produce more aldosterone. So now, aldosterone, now I know. Aldosterone. And now I know. Now I come here, I'll say the function of aldosterone. Aldosterone, yes, will make more reabsorption, or the more permeable, the nephron to become more permeable to salts. You see? Now it means that more salts will be reabsorbed. And then what happens to the level? There's increase in the level of salts in the blood, and then it goes back to normal. So everything must be linked to that. Look, normal level of glucose. You see, if there is increase here, check here, there is decrease. If there is a decrease here, check here, there is increase. So you have two places to fill in. The first one is for the gland. See, pancreas secrete insulin. You see, now here the function of the insulin. You see, glucose is converted into glycogen. So insulin is going to stimulate the liver to convert glucose to glycogen. And then what happens? The level of glucose goes back to normal. So just guys, draw this, put what you want to talk about, right increase, right decrease, right decrease, right increase, whatever. They now talk about the gland to produce that hormone, and then talk about the function of the hormone. It's done. I don't know why people, they're always talking about that this topic is a problem. Sometimes it's a problem because you don't know how to tackle it. It's a simple thing. Simple. Ne? Yeah. Just want to make things simple, because I don't want people to have problems with reading, man. Okay, diabetes. This one, please, when we ask you about the diabetes, call it diabetes mellitus. Don't say diabetes, sugar diabetes. We don't want to hear that. It's diabetes mellitus. Diabetes mellitus. If there is the pancreas cannot work properly, we call it diabetes mellitus. So please don't go without knowing this diabetes. Ne? I like it. Whenever they bring carbon dioxide, people fail, man. I don't know why. But it's the only one which doesn't have 2 ways. Ne? It's the only one which doesn't have those the 2 ways. So please at least try to master it. But the rest, you see, the same thing I talked about, normal level of water, decrease. You have here the more or increase, the pituitary gland, you see, now you function of the ADH. Simple. So you guys, this must be your guide for you to pass. Will you be doing memo for physics paper? Yes. After my live, yes, the memo is gonna be coming. Eh? Yes, the memo is gonna be coming. So around 8 o'clock. Eh? Yeah, 8 o'clock. 8 o'clock. Unless there is a change in the scale of electricity, but 8 o'clock or 8:30. Eh? Yes. Hey. And the memo for life science, definitely. Geography, definitely. Everything will do it, né? So that you gauge yourself. Né? Yeah. Anyway, so we are done with that. Skin. Skin is S. Yeah, S. So sweat, what you need to know about is sweat gland, sweat duct, sweat pore. Then what you produce is sweat. 1, 2, 3, 4. Sweat gland, sweat pore, sweat gland, sweat duct, sweat pore, and then sweat. So the S, if they ask you to label, and then blood capillaries, that's it, nothing else. So here, hot day, you'll be seeing more sweat. So please know that how to operate this. Hypothalamus is stimulated, send impulse to the blood vessel. The blood vessel become dilated. it is a mark, and this is called vasodilation. It's also another mark. Please don't skip it. And then now once it becomes what happens, more blood will flow to the skin, no, the sweat gland, the skin, yes, and then more heat is lost. But doesn't stop there. More blood is going to be sent to the sweat gland. The sweat gland becomes more active, and then when it becomes more active, more sweat is going to be produced, and then evaporation is going to lead to the cooling of the body. If they ask you during cold day, it's the reverse. Sometimes they ask you that which hormone works. They work. What is that question where they always ask you that and people go off? I think, yeah. Explain why temperature in air. No. There is name one hormone that would have the same effect on blood vessels as observed in B. Check, guys. How do I know that this is there's hot day, cold day? Check here. This thickness is big. Here is small. So this is hot day. You'll see sweat. So those are the two things we should need to look at. The blood capillaries, they are dilated, and then the sweat. Here, the blood capillaries are very small, and then they are narrow, meaning that they are small. They are below the skin surface more, and then there's no sweat. So basically that's it. And then they are saying name the hormone that would have the same effect with blood vessels observed in A. Here, like the blood vessels constrict, is adrenaline. Eh? Please don't forget about it. Yes, it's adrenaline. Tropism is the last topic, guys. Ne? And this one is also simple. Ne? What is tropism? When you talk about tropism, is a simple thing. Let me check here if there is any question. Okay, there's no question here. Right. If you have any question, guys, let me know so that I solve it down. Ne? Yes. Okay, let's go. Tropism, we say that is a simple thing, so that general function of auxins. Talk about the function of auxin. Remember, it is found at the apex, so brings about apical dominancy, growth promoter. So basically, in this, we always talk about auxins. Ne? Yes. Abscisic acid is dormancy. It's for dormancy, not dominancy. Dormancy. Someone says that you are dormant. That is an insult, man. Imagine someone says you are dormant in bed. Oh, man, you are dormant. It means that you have too much abscisic acid. Anyway, humans don't have abscisic acid. Yeah. So dormant means that you can't do. Eh? You are like a cockroaches in winter. Do you have cockroaches at your home in winter? Don't tell me that you don't have cockroaches. You have. Even I say that South Africa has a problem with the cockroaches. And in the summer, gibberellins. Gibberellins, yeah. So that's how plants also do. In winter, they have too much abscisic acid, they undergo dormancy. And then in the summer, there's too much gibberellins, and then they sprout out. Ne? Yes. So please know that. So basically, gibberellins work in summer while abscisic acid works in the winter. So gibberellins breaks dormancy, while abscisic acid basically it brings about germination, while abscisic acid brings about what? Dormancy. Yes. And the shading of the leaves. There are so many. You can read the function there. All right. Here is the last part which I need to talk about before I conclude with the graph or the interpretation. Yes. Here saying that when they shoot auxins, guys, what is important here? What you need to know that auxins are found at the tip. Auxin cause growth in shoot. In a root, it limits. So where there is high concentration of auxins, there will be faster growth in shoot, while in roots, where there is high concentration of auxin, there will be slow growth, then to be the root. So if I bring something like this and ask you that something is like this, and I say that there's too much auxin here, and ask you, is this a shoot or a root? It's a root. Why? Because the high concentration of auxin is growing slower. So it means that it's a root. You understand? Yeah. So please identify that, and then the experiments. So know this, yeah, for root and the shoot. Clinostat is an instrument which is used to remove the effect of gravity. So the plant will not bend, whether you are upside down or what, it will not bend, it will grow straight. So why? Because the effect of auxin is being removed due to gravity. Plant mechanisms. These are simple thorns. And guys, I think you have the paper. I think you have the paper. All right, let me just keep quiet there. And then I said that graphs, guys, investigation in most cases when they bring like this is independent, this is dependent. I, D, X, Y. This goes to x-axis, this goes to y-axis. Yes. In most cases, when you read here the first statement, you will see which one is that you understand. Determine the effect of distance on, you see, curvature, meaning that the distance will determine the thickness of on the curvature of the lens. The distance will determine how big is the curvature is gonna be. You understand the thickness. So please, when they ask you line graph, please title axis. Make sure the axis they have x and y. When they ask you independent reliability, validity, if they ask you in past tense, guys, always answer in past tense. If they ask you in present tense, always answer in present tense. If they say, what did they use? You tell us what they used. They used, how will you improve? You tell us you will improve by, you understand? So the English now in life science, in this section, we mark it. Unfortunately, there's nothing I can do. But the only thing I can do is to tell you exactly what happens there so that you don't make a mistake. Not everyone has a chance to get this before they write. Yes. So please make sure that at least you know that. Percentage increase, percentage decrease, percentage increase, we say that is final minus initial over initial times 100. Yes. So basically, when you get it positive, it means that is percentage increase. When you get it negative, it means it's percentage what? Decrease. Yes. So basically, guys, that is it. Make sure that you pass, you pass, you pass, you pass, you pass. I mean, I wish you the best. Unless you have any challenge. So why do you use agar jelly? Agar is like agar is like soil. So agar, the function of agar, we put maybe a growth promoter, IAA, indoloacetic acid, and then it helps to hold that. You understand? So agar is just like something which holds the what? The thing to be in place. Yes. So basically we use agar when we are culturing the bacteria. Then another one, is there anything, any other question? Guys, if you have any question, let me know. If there is no question, then I'll say that I wish you the best. I wish you the best. I wish you the best, guys. Read and read and read and read. Go through this video 2-3 times. Focus on what I've talked about. Remember what I've tried to say. If there is something I forgot, maybe I'll talk about it, or you can ask me. I'll try to help you out. If you don't have a distinction material, you can request it. It's just 20 rand, and then you get your book. Use it. At least get your distinction. See you in the paper, or see you when you're discussing about the memorandum. Yes. Memorandum will be there that day, I think around 10 o'clock. On Monday, on Friday, 10 o'clock. Monday, 10 o'clock. Did I say 10 o'clock? No, man. 4 o'clock, not 10 o'clock. 10 o'clock, I'll be sleeping. See you when you see me. See you when you see me again.
Recording stopped.